Dear students, welcome back to Applied Environmental Microbiology. In the previous lecture, we talked about exposomes and we learnt about how different research groups are doing research on better trying to understand the personal exposure versus the exposure that we would estimate from ambient air quality. Now, similar research is being done on water pollution and on basically environmental microbiology. In this lecture, we are going to briefly go through them. So, let us get started. So, coming back to the facility exposomics, that is doing that last lecture we talked about their research on air pollution. So, let us look at what work they are doing on water. Here is what they have to say. Exposomics, that is their company institute, makes use of existing short term experimental studies. So, we have short term experimental studies and long term population studies, and then they have different kind of information that they get from short term experimental studies. For example, they find out what specific chemicals and mixtures are present in water, and life course studies they understand what are the public health effects and long term exposure to different kinds of contamination. They also use information from other databases such as remote sensing and land use maps to find out what the personal exposome is, and should we validate the need for understanding the personal exposome, studying exposome instead of just what is there in my glass of water. Alrighty, and so this is one of the studies that they did in swimming pool. So in swimming pool, every time the water is filled or the swimming pool is cleaned, the water is chlorinated to disinfect the water and make sure that the swimmers don't fall sick. Now we know this thing that if there are natural organic matter, so if there is natural organic matter present in the water, it will react with your halogen with chlorine and it will create disinfection byproducts. A typical disinfection byproduct is trihalomethane. So basically we know methane is CH4, 3 of its carbon get replaced and we are left with CHCl3. Now these trihalomethanes or disinfection byproducts are not good for public health. Many of them are carcinogenic, many of them are irritants, mutagenic, teratogenic, and they are worse for ecology. So once this water is released in the environment, it is really bad for our aquatic systems. And definitely microbes are affected by disinfection byproduct. Now we also know that disinfection byproduct, beside all these problems, they also increase amount of antimicrobial resistance. The levels of antimicrobial resistance in water and wherever DABPs are exposed rises when DBP rises. So definitely we don't want disinfection byproducts, they are summarized as DBPs. Now in swimming pool, when the water is chlorinated over and over again, we develop lot of DBPs. In fact, you can smell them, smell chlorine and then smell DBPs in the water. Now the people swimming in this swimming pool are exposed to the DBPs. Now this is a part of their exposome. Now we want to know, is it affecting their health, and if it is, how much and how. So this was the study, the Piscina study, and they looked at the internal exposome. So they are not looking at external, they are looking at internal. So you can understand they are trying to do metagenomic study, proteomic study, metabolomic study, and transcriptomic study to understand how the internal environment, the internal microbial community is being affected when they are exposed to DBPs. And for this particular study, they did metabolomic analysis, and then they did PCA, which is principal component analysis, which is a statistical technique they did, which is presented here. So basically you have the exposure to disinfection byproducts. These are disinfection byproducts that they noticed, and their short term effect in swimmers because they looked only in short term. And this is untargeted metabolomics of blood samples, so they did not target, but they just did metabolomics. They did it for 3,761 people, which is quite a large number. So if they have conclusive evidence for anything, it should be statistically significant. And they noticed that they found these different disinfection byproducts in good quantities. And when they did the metabolomics, untargeted metabolomics, so they were not looking for acetate or glucose or any particular metabolite, but they did the UPLC MS on the entire metabolites that they found in the blood and serum, And on the orange what you notice are the pre-samples, so pre-swimming samples. So these is the people who came for swimming, and before they started swimming what their metabolites were, the profile of metabolites. And here you have post swimming in the blue, post swimming metabolite samples. So their blood and serum were drawn prior to swimming and then after swimming. Now we notice that there is quite a shift. The metabolites move from this region to this region, and for some of these candidates, people, the shifted metabolites is quite significant. Now here is the thing, this might be because of the exposure to DBPs, it also might be because of exercise. Once we exercise, our metabolites change. So that was one issue that this study overcame by looking at the physical exertion of people and whether they were swimming or just playing around the water, and by basically noticing their physical exertion, and they tried to delineate the effect of physical exercise versus exposure to DBPs. Now, what are the different techniques that we use when we are trying to understand the effect on internal exposome of the internal microbiome or internal community and internal environment when we are exposed to different things? We can do metabolomics, which is what Piscina study did. We can do adaptomics, we can do intermediate omic biomarkers. So metabolomics is when we take their plasma or serum samples and we analyze them through high resolution mass spectroscopy, spectrometry coupled with UPLC. So basically this is ultra high pressure liquid chromatography coupled with mass spectrometry, or maybe we might have MSMS for better resolution. And then we get metabolic features characterizing exposed groups are identified by multivariate statistics. So remember I was telling you about the exercise versus DBP. So we can do multi-component analyses and other statistical tests to find out if the shift is because of DBP exposure or if it is because of exercise. And then in this way we can also do we can identify which metabolic features. So this is untargeted metabolomics, we are not targeting a particular metabolite. We can find out which of these metabolites are significant for the study that we are doing, which of them are not. Then next we have adaptomics. In this we do untargeted investigation of internal exposome based on the measurement of complete categories of features such as protein adducts. In intermediate omic biomarkers we can look at biological molecules to understand the downstream effect of environmental exposure. These might include transcriptomics, epigenomics and proteomics. Alrighty, now let us look at this study published in 2009, health impact of long-term exposure to disinfection byproducts in drinking water in Europe. Now this is drinking water, not swimming water. So they found out that the trihalomethane, if you remember, which is basically CH(X3), where X is your halogen. Now if you remember your periodic table, the 7th column is your halogen column, which includes fluorine, chlorine, bromine, iodine, and chlorine, bromine, they are the typical halogens that we find in DBPs. So they looked at the trihalomethane exposure, what amount of trihalomethane disinfection byproduct was present in the drinking water. Now why would they be interested in that? Because the presence of disinfection byproducts in drinking water, which is definitely an environmental problem, is very strongly correlated with bladder cancer. So we want to understand how exposure to drinking water with high DBPs, high THM will result in bladder cancer. Now if the THM exposure varied from 0 to 15 up to 1000 milligram, and OR is the odds ratio, and the odds ratio for males and females were detected separately, and in bracket what you are noticing is the confidence interval. So what is the odd ratio of developing bladder cancer when you are exposed to this amount of DBP? So we noticed that it increased for both male and female as the exposure of THM increased. With males it went all the way up to 1.5 odds ratio. Now how did this study happen? This is very important, and how this kind of study, where we know now that DBPs in drinking water are bad for health, especially for men, more so for men than for women, and they might result in development of bladder cancer. Now this was their exposure study. They measured DBP measurement, they modeled DBP concentration, and they had cancer epidemiological study on bladder cancer and colon cancer. They did both cancers and they found that bladder cancer made more sense. Now they had this study of information on the kinds of cancer being detected from their epidemiological study for the region, and they knew the amount of THM because they were measuring it regularly. Now, the exposure assessment, this assessment, how much DBP people are being exposed to, and then modeling the exposure gives us, we can link it with the cancer studies, and we can also link it with the reproductive epidemiological studies, because we also know that DBPs affect the semen quality, the prematurity, birth defects, stillbirth and other problems. We can also gather data from the world water companies that supply water. Okay, what are the amount of NOM, natural organic matter in your water, what are amount of chlorination you are adding, or what kind of disinfectant you are adding, how many DBPs people are you are observing in the consumer end. So, all this information we can link with cancer epidemiological study and reproductive epidemiological study. And what we gain out of this linking is we get data, we get models, and we get expertise from different people, and we can do risk benefit analyses, and we can revise our policies. So, risk analysis will tell us, okay, is it better to allow some microbes to grow in the water so that we can reduce the level of disinfectants and thus reduce the level of trihalomethane or DBPs, or is it better to allow people to get higher risk of getting bladder cancer and other diseases that are because of higher exposure to or exposure to higher amount of DBPs, trihalomethane. So, we can do this kind of, we can answer these questions from these kind of studies. Like, should I increase the level of disinfectant enough so that people are safe from acute infectious diseases, or should I reduce it so that in chronic level they do not get exposed to, they do not get higher chances of developing bladder cancer. We can also understand different treatments. If the water is being given to different kind of populations, we can decide, okay, what is more important here, and we can also identify susceptible populations. For example, very clearly the male participants of the study showed a higher chances of developing bladder cancer than the female participants. So, once we know the susceptible population, we can do differential treatment. For example, the elderly might be more susceptible to getting infectious diseases. So, we might say, okay, boost up their disinfectant level so that they do not get infectious diseases, and the bladder cancer is way ahead in future, we will not care about much, we can leave that. Now, what this does is it helps us decide the policies, it helps us decide the water, the health and the implications of policy. And these studies also develop literature on toxicology and epidemiology, which overall help us understand risk benefit analyses, and also help us understand how our policy is affecting health and how we should change our policy. Alright, now this is a study by Dr. Emily Gardner at Virginia Tech, and she worked on human exposome framework for reclaimed water. So, now across the world, water is a very fresh water, potable water is a scarce resource. So, whether I want to use water for irrigation or for drinking or for washing or other purposes, it is rare. It is not rare, pardon me, it is scarce. Now, because it is scarce, we have the concept of reclaimed water. So, reclaimed water is basically, I have wastewater from a city, we treat it to high enough standards that we can now use it as a raw drinking water or drinking water. So, we are reclaiming the used water. Now, this can happen in two ways. We can do it intentionally, which is called recycling of water, recycled water, reclaimed water, or it can be de facto, where the waste of one city are upstream the other city's raw drinking water source, and automatically the effluent, treated effluent or untreated effluent from the upstream city becomes a raw drinking water for the second city. So, unintentional reclaiming of water. Either case, reclaiming water is a reality in India. It is rampant in India, and because of our high population density and the closeness and our also the closeness of our cities and our towns, and also because of our poor waste management or poor wastewater treatment and collection in the first place, reclaimed water is often a de facto in our country. However, much research is needed in that aspect. So, when we talk about recycled water, when it is not de facto, when it is intentional, then there are certain guidelines about what its quality should be, and even when we are talking when it is de facto, there are certain guidelines for wastewater treatment effluent. So, it should meet certain qualities, and ideally the wastewater should be treated in wastewater, but that is not often the case. So, let us look here. So, this is your wastewater treatment plant, and you monitor and maintain it regularly, and then when the water has been treated, it goes to drinking water treatment plant, it gets clean, and as it passes through your water distribution network, the microbes infiltrate the system, or they regrow, they form biofilms, they colonize, the microbial communities increase and get richer and richer. And this is also your infrastructure aging, the water age is increasing, your disinfectant is decaying, and at times when disinfectant decays we get nutrients depending on disinfectant. So microbial communities will grow, and by the time this human being consumes on the consumer end, not only the person is exposed to more microbes, some of them can be pathogens, but also the chlorine level has decreased, and it is quite possible that the DBPs are significant. Also, this person A is being exposed to environmental microbes, environmental genomes, and viruses and insects and pollution that is present in the environment. All of this together, including drinking water, air, fomites. So, fomites are surfaces which have pathogens attached to them, and if we touch them then we get exposed to it, and all of them contribute to exposome. So, now let us look at how recycled water, when it comes to recycled water, how that is potentially exposing us to different kinds of diseases. So, now the person might be exposed via touch, certain diseases you just need to be in touch with the water, contaminated water, and you fall sick. Some might be through ingestion route, some might be through inhalation route. So, let us look at different routes of exposure and what are the diseases that we can get through them. Through dermal contact, so through touch. So, this in US particularly where the study was done, snow making, reclaimed water is used for snow making, where people like to ski when the snow has not fallen. So, people will touch the snow, and that is how they might get infected. Also athletic and recreation facilities. So, if I am irrigating the grass in an athletic field, then the players and people on that field will get exposed to the microbes that are present in reclaimed water. And also because it is an athletic or recreation facility, the chances of injury are very high. So, not only dermal contact, but the blood contact can also happen, and also toilet flushing. Now, in this case, in dermal contact, we are mostly concerned with opportunistic pathogens such as Staphylococcus aureus, Pseudomonas aeruginosa, Acanthamoeba, cyanobacteria. Those cyanobacteria that produce toxins, we can summarize this as algal toxins. And then we are also interested in antibiotic resistant infection. In fact, Amy Pruden, who is the corresponding author of this paper, has done a lot of research on the presence of antibiotic resistant genes in reclaimed water, and she has shown that the reclaimed water has elevated levels of ARGs, antibiotic resistance genes present in them. So, we are also interested in, there is also probability that through dermal contact the people would be exposed to antibiotic resistant microbes. Next is inhalation and aspiration. So, let us say I am standing next to a waterfall and I inhale the water. Now, this is an issue in cooling towers, for example, many of our thermal power stations, spray irrigation, drip irrigation where we are trying to minimize the use of water, toilet flushing. Now, this is a very interesting study. In fact, at the same institute, Virginia Tech, there is one very established air pollution expert, and Dr. Marsh, and she is working now, she worked a few years ago on how the way we are flushing the toilet, not the way, pardon me, the toilet flushing might be a route for exposure to Ebola virus. Fire suppression, so you have fire hydrants, and when you are trying to throw the fire, you are aerosolizing water. Car washing, also shower. In this case, we are concerned about opportunistic pathogens such as Legionella pneumophila, which causes mild lung infection, Mycobacterium species, which also includes the tuberculosis mycobacteria. Pseudomonas aeruginosa causes really bad skin infections, Staphylococcus aureus also. Nigella fowleri, if you remember from your previous class, is the brain-eating amoeba, and then algal toxins, antibiotic resistant infections, fecal pathogens also, and disinfection byproducts. Now, if you look at nasal aspiration, now this is more in concern in recreation, in potable use when I am drinking the water. So, if I am drinking, I by mistake aspirate it through nasal route, or I am doing sinus irrigation, which many people in India do actually. It is called neti pot technique for cleaning the sinus. So, in this case, the major danger is of Nigella fowleri. Now, eye and ear contact, if I am washing my face or the water enters my ear, this might happen during recreation, like let us say I am swimming or just playing with the water, or direct potable use, I am drinking water, I am splashing it on my face. And then in this case, I am interested in Acanthamoeba keratitis, causes horrible infection in the eye, and Pseudomonas aeruginosa. Again, then we might have colonization and delayed infection, and there are various ways of getting them. Here again I am interested in antibiotic resistant genes, infections, Pseudomonas aeruginosa and Staphylococcus aureus. Now, if you look at our drinking water and our reclaimed water, in this study they highlighted how the recycled water that is allowed in US for different purposes, whether it is private and urban irrigation, indirect environmental reuse, differs in the quality from the drinking water. For example, the COD of drinking water has to be very low compared to the recycled water. Also the pH is similar, but the dissolved oxygen is very high in drinking water versus the recycled water that is used for private irrigation and direct environmental reuse. Suspended solids are not undetectable, whereas there is some suspended solid present in both. Again, the chlorine residual is very low for recycled water, but it is quite high for drinking water. Again, the TKN is very high for the recycled water, and the standard for this recycled water compared to drinking water. Same is true for ammonia, nitrogen and total phosphorus, iron and zinc. Well, zinc is comparable in all. What is interesting is that some pesticides are allowed in private urban irrigation, recycled water used for public purposes, irrigation purposes and direct environmental reuse, but none of them is allowed for drinking water. Also, fecal coliforms are allowed when they are used for direct environmental reuse, reclaimed water, but definitely not allowed in drinking water. So, even if I take this direct environmental, the water that is good enough for direct environmental reuse and try to use it as a drinking water, it requires further treatment. The other thing that this paper talked about, this is study from, this is information from other research, but what they talked about was, in the blue here we have drinking water and in the purple here we have the reclaimed water. So, they noticed that okay, the certain parameters of drinking water and reclaimed water are quite in agreement with each other. For example, AOC, DOC, TT, HMPF, MFP, THAFP, they are quite agreeable with each other, and we also measure them like dissolved organic carbon, assimilable organic carbon. But then there are other things like dissolved organic nitrogen and estrogenic activity, so endocrine disruption, that are present in the water and that are much higher in reclaimed water. So, as we reclaim the water and continue recycling it over and over again, they are likely to increase more and more, and thus they will affect microbial growth, algal growth when they are thrown in the environment, and the health of the people. And this is again where the this is another example where the exposome study helped us identify that okay, this is what people are getting exposed to if they are drinking reclaimed water, the amount of DBPs, the amount of DON, the amount of endocrine disruptors, and now we can do a health study and understand the health risk assessment. Now, this study also looked at advanced treatment processes that can be used for getting rid of the contaminants. They looked at ozone H2O2 oxidation followed by BAC, which is biological activated charcoal reactor, and the intended use was piloted indirect potable reuse, and they noticed that this one had more than 90% average removal in 21 of 31 targeted trace organic contaminants and hormonal products, which are endocrine disruptors. Also, the BAC achieved higher than 95% removal of all targeted contaminants except benzophenone, and then there was a high degree of microbial inactivation, so microbial presence was not a problem. But it released a concern because the AOC is increased, which will increase the microbial growth potential once the oxidants have died out, have been consumed. Also that the fluorescence excitation emission matrix, so just fluorescence microscopy showed that the transformed organic matter, the organic matter had been transformed. Then they did standalone BAC, so remove the advanced oxidation part, just standalone biological activated charcoal, and they wanted to do this for removing DOC, dissolved organic carbon and nitrogen, and they noticed that the removal rate of DOC reduces after the breakthrough point is reached. Also 50% of total nitrogen was removed, and then they did ozone peroxide, again same thing, and RO, which is reverse osmosis, and they noticed that because of high hydroxyl radicals in wastewater effluent, the removal was similar to ozone and ozone peroxide, they were very similar. They also noticed that a lot of NDMA was developed in water, so that will raise concerns on when we are thinking of future reuse application. Also, they did ultrafiltration followed by RO followed by UV treatment, and they noticed that 13 out of 291 targeted compounds were detected after UV and after RO. So, see, we notice that none of our techniques, even advanced oxidation and advanced treatment techniques, are foolproof. So, if more studies are important, more studies are required to see when these compounds are still present in water, how they affect health, and how much reclaiming of the water is permissible and how much is not. Now, this is a very beautiful diagram again from Dr. Garner's paper, and this is a cross section of a pipe, and if you notice here in the pipe we have biofilms growing, there is some extracellular DNA, there is biofilms are protecting pathogens, and we also have microbes that have developed phagocytosis resistance, so no amoeba can eat them, protozoa can't undergo phagocytosis for them. There is oligotrophic growth, so microbes are growing in scarce nutrient environment. This horizontal gene transfer which is a nightmare for antimicrobial resistance. There is also resistance to disinfection. The amoeba can form a cyst around the pathogen, so these are pathogens of concern. So, pathogens may find home in the biofilm, they may find home in the inside an amoeba, and they refuse to die. The amoeba might cyst them, and then there might be selective pressure from heavy metals, from antibiotics, from disinfectant and DBPs, disinfection byproducts. All of it will enhance the antimicrobial resistance. So, antibiotic resistance, antimicrobial resistance will increase. And the way we measure antibiotic resistance, because of this gene transfer and how important the genes are, is by measuring the antibiotic resistant genes, ARG. So, the ARG genes, now these bacteria are not necessarily pathogenic. They might be pathogens. If they are pathogens, we do not want to get exposed to them, but let us say they are not pathogenic. Let us say in our nose or in our body we already have Staphylococcus aureus present. Now, if the Staphylococcus aureus overpopulate, we will have a staph infection, and this is susceptible to antibiotics. But now these bacteria have developed antibiotic resistance, and when I am exposed to them, then the ARG can transfer to pathogenic bacteria. So, these benign microbes can transfer them to, these benign microbes will transfer the antibiotic resistance to pathogens, and now this Staphylococcus aureus could be resistant to microbes such as is in case of MRSA, MRSA. Now, the other possibility is also delayed infection. For example, I sneeze and I get wounded, or this now MRSA that has resistant microbes in my nose are exposed to other people, and now the wounds can get infected really bad. So, dear students, this is all for today, the importance of exposome. So, we want to look at all what is happening, not only what are you being exposed to, but we also want to look into what is happening in the environment from which your exposed microbial or chemical contaminants are coming, and what kind of problems we are facing and we might face in near future. And this is all for today. In next class, in next lecture, we will go more into antimicrobial resistance, which is quite a burning topic, and it is very important in our country for us to rein antimicrobial resistance, to put an end to it. So, this is all for today. Thank you very much. See you later.